Hello everyone, welcome to BioBoost. So today we will take over another organelle, which is the nucleus. Nucleus, by definition, we know it is the control center of the cell. It is a control center of the cell. Also, it has all the information in the form of genetic material. We know in the form of DNA, it has all the information needed for the cell to function. Eukaryon, we see in eukaryotic cells. So the term eukaryon means true nucleus. Means eukaryotes has membrane bounded nucleus. We go to the next part, that is the functional part of nucleus. Then we see that nucleus plays the function of storing genetic material first off. It has all the genetic material storage. It controls cell division. Then it directs functioning of cell and provide template for RNA protein synthesis. Means during DNA replication, DNA translation and everything, it provides the functioning of cell. It provides a template for RNA protein synthesis. Then we see that it physically separates DNA from the cytoplasm's complex metabolic machinery. So, because in nucleus we have DNA, so it's basically separating cytoplasm from the DNA. Also, nuclear membranes serve as boundary. Now we see the components. What all components are there in nucleus? So we see that nucleus has a nuclear envelope. Here you can see in this diagram, this double layer outside of nucleus is nuclear membrane here, or you can see here, this is the outer membrane and this is the inner membrane. So these two membranes together make the nuclear envelope. So the first component is nuclear envelope. In nuclear envelope is pore riddled, mean these minute pore like structures are there in nuclear envelope. So it is pore riddled. Next component is nucleoplasm. Nucleoplasm is the fluid interior portion, means the fluid present between these two membranes is the nucleoplasm. Here you can see it's the fluid interior to this portion. Then the third component is nucleolus. Nucleolus is dense cluster of RNA and proteins, means it is made up of RNA plus proteins. So here you can see nucleus present in middle of the nucleus and it comprises of RNA and proteins. Next, last component is chromatin, which contains all the DNA, all the genetic part of nucleus. So it comprises DNA and proteins. So when in MCQ type questions are asked in competition exams, so you will be asked nucleus and chromatin, the component part, then you have to remember that RNA is present inside nucleolus and DNA is present inside chromatin. Next, when we explore more about nuclear envelope, then we see that it is composing of inner and outer membrane with perinuclear space. The space between these two membranes is called perinuclear space. It is almost of 7 to 8 nanometer thickness and trilaminar appearance. Next is inner membrane lined with fiber network, means nuclear lamina. The inner membrane, here it is the inner membrane, so nuclear lamina is the more inner membrane to the nucleus, so it is intermediate filament protein called as lamins. It has intermediate filaments which are known as lamins. Then nuclear lamina support to nuclear envelope and attachment sites for chromatin. So here you can see these thread-like structures are chromatin, so they are in direct contact with nuclear lamina. So basically nuclear lamina provides support to nuclear envelope and also provide attachment sites to chromatin fibers or chromatin. Next we see the outer membrane. Here this one outer membrane is continuous with ER. When we see this diagram, then here you can see the outer membrane of this nucleus is in continuation with ER. This is ER, so the nuclear membrane, outer membrane is in continuation with ER. Next is outer membrane studded with ribosomes, means protein synthesis. So because it has ER, it is having ribosomes on it, which are sites for protein synthesis. Next, perinuclear space, which is 20 to 40 nanometer, is in continuous with cisternae of ER. This here you can see, on this you can see outer membrane and this one is inner membrane. So in between these two is perinuclear space. Then we see the nuclear pore. Here you can see the nuclear pore. So it is the most distinctive feature of nuclear envelope. It has small cylindrical channels which are in direct contact between cytosol and nucleoplasm. Means here it is nucleoplasm and here it is cytosol. So it is in direct contact between cytosol and nucleoplasm. It is readily visible, freeze fracture microscopy, through that you can see it easily, and the density of it is depending on cell type and the activity it is performing. Next we see nucleolus. So what is nucleolus? It is the ribosome factory. It is the ribosome factory. It has large and prominent structures. It has large prominent structures. It does not have any membrane, means this part nucleolus does not have any membrane. Next we see the component part. So nucleolus contains fibrillar component. By fibrillar component we mean it has DNA plus RNA component of ribosome. Ribosomes which are present on ER contains DNA and RNA. So the fibrillar part of nucleolus contains both of these. DNA carries genes for rRNA and RNA is simple ribosomal RNA. It is the place where transcription is going on. Okay, next in next video we will go to the granular component and discuss the same. So this is it. Thank you.